So I'm going to be talking about data visualization because I think at the sort of center of any effort to understand complex data sets of the kind that neuroscience experiments are generating has to be good visualizations. And in the kind of spirit of this conference, the argument I'd make for that is that good visualizations allow you to leverage very powerful, very large neural networks, a particular instance of which I'm showing here. Many of you will probably recognize this as a circuit diagram of the primate visual system. And so our visual systems are extremely powerful, and they're very good at things like pattern recognition and detecting structure in data, which is exactly what you want to do when you're trying to understand a data set like recordings from neurons. But the problem is that the sort of input channels to the visual system are very constrained. So you can't just take raw data and dump it into your eyes, onto your retina, and expect to get any insight from it. So I work with humans, and I generally am using fMRI data. I'm showing an example of some kind of a raw data that I work with here. And while any insights that we're going to be able to get from this data are in some sense encoded here on the screen, they're not presented in a way that your visual system is able to make anything of it, right? And this is just about maybe 10 seconds worth of data. We collect hours and hours of data and want to do something with it. And so the approach you should take when you're thinking about putting together visualizations and using visualizations in an analysis workflow is something like doing feature engineering for the particular network that you're working with, the visual system. And in particular, the kind of principles that I think guide this are you need to aim at some sort of abstraction of the kind of data that you have, transforming it from the raw data, whether it's something like MR, signal intensity, or calcium images, or spikes that you're getting off an ePhys recording, and abstracting it in ways that you can make sense of it with your eyes. And then and you do that basically through the kind of parallel processes of aggregation, of grouping sort of similar kinds or parts of your data set that you think are similar together, and estimation, right? Using statistics, using models to extract structure from your data and present it in that way. And so in particular, I'm going to be talking about a software package that I've been working on called Seaborn. So Seaborn is a Python library. It runs on Python, and it's built right on top of Matplotlib. So Matplotlib, if you've done any sort of data analysis in Python, you've probably used Matplotlib. And so you know that it's a great package. It's very flexible. Basically, anything you ever want to draw onto a computer screen can be drawn with Matplotlib. But the flip side of that is that if you've used it, you probably also know that it can be somewhat cumbersome to draw useful things. It can require a relatively large amount of code to put together a visualization that is informative. And also, in the past couple of years, Pandas has really become, so Pandas is a library that offers, among many other things, a data frame object, an object that structures both numerical data, but together with semantic information about what that data is, what it corresponds to in the context of your experiment. And Matplotlib doesn't really know anything about Pandas, and so Seaborn brings these two together. And in particular, it offers, on top of Matplotlib, a very high-level interface for drawing statistical graphics, for drawing visualization of data in ways that will allow you to make insights from it. Particularly, insights using your visual system, detection of patterns and structure. And so because it's built on top of Matplotlib, Seaborn generates basically static figures. It's not interactive in the sense that we're going to hear about with some of the libraries that target HTML. But the hope, the ideal at least, is that the interface is expressive and concise enough that you can achieve what I like to think of as reproducible interactivity. So interactivity in the sense that you can generate a figure to answer a question that you have about your data at roughly the speed at which you can think of those questions. But then it's reproducible in the sense that it's all in text, it's all in code, so you can work through an analysis, go do something else for a week, come back and know exactly how you made a particular figure, which I think is really important in science where projects persist for a while. Okay, so for the rest of the talk, I'm going to show examples of the kind of plots you can make in Seaborn, how you do them, some of the ways you should think about approaching your own data if you want to use Seaborn to take a look at it. So working with the kind of data that I showed at the beginning, this raw data set of bold time series data. This is data from humans while they're lying in an MRI scanner doing stuff, and we're recording from their brain. We're recording blood flow. And so the first kind of visualization I want to show is a visualization that it basically represents all of the information in that raw data set, or at least all the interesting information, the recordings from the brain part of the volumes that we acquire. And so there's a lot of data here, but at the same time it's been abstracted, right? So this no longer looks like a brain. And I've accomplished this by aggregating, so breaking the brain, the cortex up into about 60 or so parcels or regions of interest and averaging time series data within them, and then aggregating in the time dimension by computing each cell here shows a pairwise correlation of the time series data. And so this reduces the data quite a bit, reduces the data sort of using knowledge that we have about how the cortex might be organized. But what we gain from that is we're able to extract some structure from this, right? So we can see that you can basically reproduce known sort of large-scale network structure in the cortex that even when subjects aren't really doing anything, they're just lying in the scanner, some parts of the brain tend to correlate with other parts of the brain and tend not to correlate with even other sets. Okay, so this kind of visualization isn't that hard to make in Matplotlib, although some aspects like dealing with the labels can be kind of annoying, and the default color map sucks. So in Seaborn, this is just called a heat map. So you just call the heat map function, you pass in a pandas data frame, and in fact I'm just using, the pandas data frames have good methods for doing various transformations here, just computing the correlation matrix kind of on the fly. And then it takes all of the semantic information that's part of that data frame and knows what the rows and columns correspond to, puts them on the side of the figure, and then it sort of automatically detects that this is a correlation matrix, and so it should use a color map that goes from sort of cold colors to warm colors centered on 0. And so color is obviously a really important aspect of the human visual system. It can either help reveal patterns or obscure them if it's used improperly. So using sort of good default colors and color maps for heat map data is important, but also defaults are boring. So a big part of Seaborn is the ability to make custom color maps. And in fact, this is in the development version, but using some of the IPython interactive machinery that Fernando was talking about last night, so you can actually sort of play with all the various parameters on some of the more complicated color map generating routines and come up with custom color maps that you can use for your own data. Okay, so heat maps are a nice way to extract really high-level structure, but there's a lot of aggregation going on there. If we want to know kind of more about what's going on in each of those little cells where the only information we have about it is a color value, we have to make some sacrifices. Let's pick one of those networks and zoom in on it, and then that's a trade-off we make. But on the other hand, we can actually see what the underlying data that's generating this is, also just looking at a relatively small slice in time. So here what I'm showing is, so each of the columns and rows here is one of the columns and rows in the heat map. It's average data from some region of interest. And then the scatter plot is, shows the same relationship that you got the color map value from. But you can see that there's kind of more structure here, in particular that the time series are making these sort of swooping trajectories through this joint space. On the diagonal, I'm plotting a kernel density estimate, an estimate of the univariate distribution, so you can get a sense for sort of how these variables are distributed, whether the statistics that you're going to use or actually make sense to for this kind of data. Okay, so this is a very informative, if somewhat more narrowly targeted visualization. How do you make this? Pretty simply, this is called a pair plot. You just pass in a data frame. It extracts, by default, it extracts any of the numeric columns and then shows all of the pairwise correlation or pairwise relationships as a scatter plot. And here I'm just saying on the diagonal, instead of showing me a histogram, show me a kernel density estimate, a sort of non-parametric estimate of what the distribution looks like. Okay, so let's try to emphasize this sort of trajectories through time by mapping time with a color here. So this is basically the same plot, but emphasizing something else about the data. And I want to show this to demonstrate a kind of more general point. A lot of these Seaborn functions like pair plot, which generate very or reasonably complex figures, are actually built on top of class objects, in this case what's called a pair grid, that you can use directly if you want to make this kind of visualization but do something that isn't necessarily offered by the API of the function, which is generally somewhat more targeted and constrained. So here, generating the pair grid object from the data frame and then just saying map, a plotting function. So here just the matplotlib scatter plot function, and that lets you color each point separately. So this map concept of just applying a plotting function to each facet or to each cell in this grid is a kind of basic pattern for using some of the more complex Seaborn objects. Okay, so another alternative you might want to take, moving from that heat map to a more informative visualization, is instead of narrowing your focus, is doing another step of aggregation. So here, aggregating with each of these networks we can identify across the constituent nodes of that network, and then plotting the distribution of correlations with all of the other nodes in the brain as what's called a Violin plot. So this is also based on a kernel density estimate. This is what was on the diagonal of the pair plot I was showing you before. And it gives you some idea of what the structure of the kinds of correlations that each of these networks have with the rest of the brain. So this is called a Violin plot. It understands these panda data frames, and in fact, it understands this sort of long form or tidy approach to structuring data where you pass in a column that just has all of the correlation values and then say group by a column that has all of the network identification. And then also you can sort of tweak the kernel density estimate and set colors nicely. Okay, so let's zoom in even further on a particular relationship. And in fact, now I'm using the output of, there's been another step of estimation done elsewhere in Python where we fit a linear model to each voxel and obtained a sort of summary statistic or several summary statistics for each voxel, just expressing how that voxel correlates with different aspects of a behavioral task. So when, again, we've made a trade-off. Now we're looking at a much narrower slice of the data set, but we've sort of freed up parts. So we freed up space on the visualization to show more information about it. So here we're looking at the joint distribution as a scatter plot. So that's simple but informative. And then also showing the marginal distribution of each of these variables as a histogram. So again, you can get a sense for what do each of these variables look like in isolation, what do they look like together. So again, this is quite simple to do. This is a function called joint plot, and you just say the names of the columns in the panda data frame, right? So it's very semantically informative code. Instead of passing in a NumPy array and saying, show me the 4th column and the 6th column, it's show me the columns that have this kind of information, and then you get something hopefully informative in just a single line of code. Of course, there are other ways you might want to emphasize the relationship between these two variables. So for instance, you might want to fit a regression function and ask how much the variable on the y-axis is changing as a function of the variable on the x-axis. How strong is this relationship in a kind of different sense? That's easy to do. You just say kind equals reg, and it does the estimation behind the scenes using stats models. If you have a lot of data, scatter plots can sort of quickly grow uninformative, and so again, you want to perform an estimation step here, estimating a two-dimensional kernel density estimate, so a two-dimensional estimate of the joint distribution. Again, that's very simple. It's just kind equals KDE. So there's a few other options here, and then also much like pair plot, joint plot is built on top of an object called joint grid, which exposes a sort of more intermediate level API to build this kind of figure with maybe kinds of plots that aren't sort of built in as defaults. Okay, so again, focusing on this case where you kind of have a scatter plot and a regression model. When you have large data, large data is interesting not intrinsically, but because it allows you to ask questions about interactions in your data set, how particular relationships that you care about change as a function of other variables in your data set, right? So this is a really key thing, is ask, well, does this move around if I condition it does this relationship move around if I condition it on different levels of other things I've measured or manipulated? And so here, fitting the same kind of regression to this scatter plot, but doing it separately for different hemispheres. I mean, this isn't particularly interesting data, but it gets the point across. And so this kind of basic thing, fitting linear regression models conditioned on other variables in your data set, are done by a function called lmplot for linear model plot. So here again, lmplot, the x variable, the y variable, and then the hue variable. So say condition on this, the hemi variable, and plot them in different colors so that you can tell them apart. You can ask somewhat more complicated questions this way. So in addition to faceting on a hue variable, you can actually create multiple instances of the same kind of plot showing the same relationship, but for different parts of your data set. And again, this is just applying a call for a column, mapping this to another facet. And if you have a variable with a relatively large number of levels, you can sort of wrap so that you can see this relationship on every subset of a relatively large variable or moderately large variable. Okay, so when you're talking about relationships between different kinds of variables, obviously scatter plots aren't going to work for everything. So in neuroscience, obviously time is a really important kind of variable, and you don't necessarily want to make a scatter plot. So there are other functions that kind of know about things like time series data. So here, plotting just estimated hemodynamic responses. This is what blood flow looks like when you do kind of various cognitive tasks. So this is what's called TS plot for a time series plot. And again, you're passing in the raw, not maybe raw, but preprocessed, but a full data set, a data set that has each observation, or an observation at each time point for each here subject, for each condition. And you're saying it's, plot this bold data, the column and the data frame. And it, behind the scenes, Seaborn will do the aggregation and then estimate confidence intervals and stuff like that so that you can get a sense for how variable this relationship is across whatever unit you sampled the data at for here subjects, but it could be something like neurons or brain regions. So obviously it's kind of most informative to collapse over these sampling units, but it's also really important to look at the data in as raw a form as is informative, if nothing else, than to make sure that nothing's gone wrong in whatever transformations and preprocessing steps you have applied. And so here, in addition to plotting the sort of average time series from before, I'm also plotting in the background each subject's estimated time series. It's quite a bit more noisy, but you can get a sense for where the basic structure is coming from. And that's just setting one of several error styles here, saying show me a trace for each of my sampling units. Sometimes you want to show relationships between a continuous variable and a discrete variable. Here, the discrete variable is each of these RYs from before. And in this case, something like a bar plot or a point plot is going to be more informative. And so this is using a Seaborn function called factor plot. So factor just in the kind of R sense where it's a discrete variable. Showing the x and the y factors, and then again, you're passing in a full data set here with an observation, with each of the observations that you've measured, and the function behind the scenes does the aggregation, bootstraps to get confidence intervals, plots the error bars without you having to ask it to do anything in particular. And then it returns, so factor plot and LM plot are, like some of the other functions, built on an object called a facet grid, again, one of these sort of intermediate level objects that it returns, and this object exposes, again, an API for manipulating things like the aesthetics here, just rotating the tick labels so they're not overlapping each other. Again, you can use this object directly if you want to make make one of these kinds of plots in a way that isn't anticipated by the functions that exist. So here, similar to a plot I showed before where there was a regression estimate on each of the facets, but here showing a kernel density estimate of just univariate distributions split by two faceting variables, so the column facet variable and the hue facet variable. And so here's how this gets made. You instantiate a facet grid object with the data frame that comes into it, tell it how to kind of organize itself in terms of what variables you want to condition on. And then you map various plotting functions across it. So you can pass in matplotlib functions directly. It knows what to do with them. Seaborn functions, this KDE plot function. And then in various ways, deal with the aesthetics and make tweak the plot if you want to, if you don't like exactly what the defaults give you. OK, so that's all I'm going to show today. If you want to know more, you can check out the docs. There's an example gallery with kind of short scripts that illustrate a lot of the different kinds of plots you can make, and longer, more verbose tutorials that walk through all the various options of these functions, some of which can get quite complicated. And if you want to check out the code, it's on GitHub. Thanks.